All right, so welcome to our virtual Bio 240 lab. We're making it up as we go. Kurt is helping me, so some of you have him as your lab instructor, and some of you have me, and the other lab instructors, they will be emailing you sometimes with some updates. But for now, we're doing a crash course in some of the lab information that we want to make sure that you have. And some of it's a review, especially for those of you that actually got to do lab this Monday. You already did the reproduction in seed plants. Some of us didn't get that, so this will be the review for those of us that didn't get it as well, or not a review, but actual information. So let's go back to plants in general. We started with our primitive non-vasculars. The hepatocophyta were the liverworts, the bryophyta were the moss, and those are both gametophyte dominant. So remember, gametophyte is haploid and make gametes. And then we move to the vascular tissue. Starts with the lycophyta. That's also where you get the roots, the leaves, the stems. And now we have sporophyte dominance from here on out. So lycophyta are the club moss, and we had Selaginella and Lycopodium. And the Selaginella were the ones that were heterosporous, the Lycopodium were the ones that were homosporous. You saw slides of those, super cool slides. And then we moved on to Pterophyta, and those are the ferns, the whisk ferns, and the horsetails. And you saw the gametophyte for the ferns, making the gametes, the sperm and the egg. And then for the horsetails, it was the strobili. So you were seeing the spores inside the strobili. Then we're moving on to the gymnosperms and the angiosperms. So these are seed plants, and some of you have already done it in lab, and some of you have not. So for everybody, it's just good to review anyway. So for the seed plants, we've got the naked seeds, which are the conifers, the gymnosperm, the ginkgos, the cycads. Those are the ones that you need to remember for the gymnosperms. And the angiosperms are enclosed seeds, meaning anything with flowers. And we're going to go around campus looking for flowers today and looking for pollinators. So we'll get to that. So all plants have sporic meiosis. That's the life cycle. When we were in fungus, we talked about zygotic meiosis. When we were in algae, we did zygotic, sporic, and gametic meiosis. But remember, for plants, it's all sporic meiosis. And that means half and half diploid, half haploid. Starting with the sporophyte, it's going to make spores by meiosis. That's the only time we're going to have meiosis. The spores are 1N. So remember, meiosis cuts the number of chromosomes in half. So we're going to start with 2N, go down to 1N. The spores are going to grow into gametophytes by mitosis. Everything from here on out will be mitosis. The gametophytes make gametes, so always remember from the name, sporophytes make spores, gametophytes make gametes. The gametes, sperm and egg, once they unite, that's fertilization, will become a zygote. The zygote is 2N, and that grows by mitosis into the sporophyte. So that's the life cycle that you're going to remember all the time for plants. Sometimes the main plant that you see is a gametophyte, like for the moss and for the liverworts. And for all the rest, the dominant plant is the sporophyte. Like the big tree that you see is a sporophyte. The gametophyte is in the cone, so you're not actually going to see it unless you're looking at a microscope and looking in the ovule or looking at the pollen. So let's go over, let's shift on over to our gymnosperms. Does that work, or should we move it a little bit this way? Okay. And for the gymnosperms, we're going to start with our sporophyte. And the sporophyte is maybe the tree, the pine tree, let's say, that has two types of cones on it. It's got an ovulate cone, which is a female cone, and a staminate cone, which is the male cone. So you do need to know those terms. Inside the cone, the sporophyte is making spores. And by meiosis, but you're not going to see those, not even in the slides. The spores are going to grow into the gametophyte. The gametophyte is what you see in the slides. So inside the cone, I'll show you actually slides after we get through with this. There will be ovules near the base of the scale of the cone, and in a staminate cone, there will be pollen. That's the microgametophyte, so that's the male gametophyte, versus the ovule, which is the megagametophyte for the female. All right, so inside the ovule, so that's our sporophyte, it is making gametes, because it's a gametophyte. And for pine, I put 2, because there's usually 2 eggs in the ovule of a pine cone. So I want you to know the parts of an ovule, and you're going to remember that that's the megagametophyte. And there's an opening called the micropyle. I'm sorry, you're going to have to see all my really messy handwriting and scribbles. But the micropyle is where a sperm would enter, and it would only fertilize 1. So the gymnosperm does not have double fertilization, it just has 1, and it's just going to fertilize an egg. Meanwhile, also in this ovule would be megagametophyte tissue, that's here, and that's 1N, because remember gametophytes are 1N. And around the ovule is the integument, so that's the layer around the ovule, just 1 layer of integument. Once fertilization occurs, so we've got our pollen here with the sperm. Sperm is not flagellated because it's going to be dispersed through the wind. The sperm is going to find the base of a cone and make its way, hopefully, into an ovule. And that becomes the seed. Once the sperm fertilizes an egg, now we've got a seed. To the seed now, so once fertilization occurs, we have the gymnosperm seed, so like a pine seed, which you can eat. And that still has this megagametophyte tissue, which is still 1N. It has an embryo in the center, which is 2N. And now the integument that was around the ovule has become the seed coat. So you need to know the parts of the ovule, which is the megagametophyte, and as well as the parts of a seed after fertilization. All right, so remember that the pollen is microgametophyte. It's made inside the staminate cone. And we're going to show you some slides of what these look like. I'm sorry about my scribbles, but you'll see the slides later. All right, so we're going to move on to angiosperms. Angiosperms are flowering plants. So anything with a flower is an angiosperm. Sometimes you don't even notice it's a flower, like a grass. That's an angiosperm. Sometimes you don't see the flower, but they do have them. So let's start with the sporophyte. And actually, we have our handy-dandy, I'm going back. Our handy-dandy model here. So here's our flower. And you do need to know the parts of the flower. In fact, I want you to draw it in your lab notebooks. I'm going to tell you what you need to have in your lab notebooks for when we come back from break. And one of them is going to be a drawing of a flower labeling the parts. So we've got sepals are for protection of the bud when it's closed up. We've got petals that are for protection of the reproductive organs as well as for attraction of pollinators, because now we're going to have pollinators helping disperse the pollen to get the sperm to the egg. We've got the male portion have filaments and an anther, and the anthers are where the pollen is made. So that's where the microgametophyte will be made, in the anthers. And then the female portion will be the ovary, which contains an ovule or multiple ovules. In this case, I'm just showing one. A style is the part that's heading down to the ovary, and then the stigma. I always think of sticky stigma because the pollen's going to land on top there. It's going to send a pollen tube that it generates down the style, and the 2 sperm that are discharged from 1 pollen will have double fertilization. 1 will pollinate an egg, and 1 will pollinate the polar nuclei. So we'll get to that. That's going to help make the endosperm of the seed. But I do want you to know all the parts of the flower. You're going to draw it in your lab notebook, and you're going to list the functions as well. All right, so let's go to our diagram here. All right, so can they see this as well? Okay, so with our flower, sporophyte is 2N. We got to make some spores. So you're not going to see the spores in the slides, just know that they're happening. So for the mega spores, we're going to make them in the ovary. We're not going to make them, they're going to make them. And the microspores are going to be made in the anthers, right? Because that's where the pollen is going to be made eventually from the microspores. So remember, the spores are going to grow into the gametophytes by mitosis. So the flower is making those spores by meiosis first. Again, we're not looking at them, but they will be here. And then the megaspore will grow into the ovule, which is the megagametophyte with the egg, and the microspore will grow into the pollen which is the microgametophyte with the sperm. And there will be 2 sperm per pollen now, okay? So let's see, we've got our parts of the flower I already did. Let's do some double fertilization with our ovule. So you do need to know the parts of the ovule, which are slightly different than the ovule for the gymnosperm. So for the angiosperm, it's more evolved. It has more cells involved and more nuclei. So we've got 1 egg, 2 synergids, 2 polar nuclei, and 3 antipodal cells. So that is 7 cells in total if you count this whole big ovule, and 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8. So 7 cells, 8 nuclei in this one ovule. The sperm, 2 sperm, will come in through the micropyle. So there's still a micropyle. 1 will fertilize the egg, and the synergids, the job of the synergids is to guide one of the sperm to the egg. The other sperm is going to come and fertilize where this polar nuclei is, so basically this whole area. And these are going to unite with that sperm nuclei to make 3N tissue, which is going to be called the endosperm. So we've never had 3N, we've always had 2N or 1N. This is the only place you're going to have triploidy. And now that we've fertilized, double fertilization, the egg and the polar nuclei, we're going to end up with our seed. And just know the antipodal cells, they're for some nutritive functions. They kind of balance out the ovule, but mostly I want you to know what the synergids do. They're to guide the sperm, as well as that the polar nuclei will be fertilized to make the endosperm. The endosperm in the seed is what's going to nourish the embryo. And I should have mentioned that for this seed as well. So for the gymnosperm, it's megagametophyte tissue that will nourish the embryo. So know the difference, because we often ask that on a test. So this is megagametophyte tissue is 1N, endosperm is 3N. Both of them are there to nourish the embryo, which is always 2N. Now, for angiosperms, they have 2 layers of integument in the ovule. For the seed, it will have a thicker seed coat, so it's more protected for the angiosperm seed. And we are going to talk about dispersing seeds a little bit later when we talk about our lab 14, has to do with fruit. So some of the fruit that you eat is a ripened ovary with the seeds inside. So you should know the difference now between an ovary, an ovule, and a seed. So an ovary houses the ovule. The ovule is the megagametophyte, which becomes the seed after fertilization. And then it can be dispersed when an animal eats the fruit, the ovary, and disperses the seeds that way. Or sometimes the fruit pops open. If it's dehiscent, it can just pop open and disperse seeds that way. So there's multiple ways that seeds can be dispersed. We'll talk a little bit about that later, but just know that the seed is what is going to germinate into our plant, the sporophyte. So our seed would be the beginning stage of our sporophyte. All right, I think we're ready for some other stuff. So now we're going to scroll through some of the slides that some of you already saw on the Monday lab and some of you did not get to see. And I just want to let you know that you will not have a practical for the plant portion. So this is just so that you get to see what's inside the cones and the ovaries and the anthers. But we won't be setting up microscopes for you for this portion because we'll just do a written test. And you'll still be doing your presentations when we get back from our break here. But the slides for now are just for you to enjoy. All right, so here is our ovulate cone. So remember, an ovulate cone is the female cone on a gymnosperm, and we're going to look for some ovules. So these are all scales, and the ovules are at the base of the scales. And at this point, they are starting to develop, so they would be developing from the megaspore growing into megagametophyte. So it's happening here, we're still developing, more developing, and now we've got our ovule. So with our ovule, there, remember I said there's 2 eggs, it's a pine ovule. The micropyle would be here. The integument is around here. We've got the megagametophyte tissue, of this ovule. Here's another one, just a close-up. You can see the micropyle and the two eggs. So the eggs are the gametes made by the megagametophyte. This is the seed of this ovule after fertilization. So now we've got our embryo is 2N, our megagametophyte tissue is still 1N, and the seed coat would be around here, around the outside from the integument. This is the male cone. So the male cone is the staminate cone, much smaller. When we're out on our walk, we'll point out some female versus male cones. And inside the scale, it's making the pollen from the microspore that, remember, what makes the microspore first by meiosis, and then the microspore develops into the microgametophyte, which is the pollen with the sperm inside. So it looks very different from that female cone. And once that pollen is dispersed in the air by the wind, it will hopefully find that female cone and fertilize some ovules. So this is just a close-up looking inside to look at the pollen inside. And you can see some of the nuclei for the sperm inside as well. This is a bud, lily bud. And it's got the 4 whorls. If you notice in the lab manual, it talks about the whorls. Those are all modified leaves. So the outside would be the sepals, the next layer would be the petals, then we've got our anthers making the pollen, and our ovary with the ovules, the style, and the stigma. So that is a bud. Now we're going to look inside the ovary for ovules. So remember the female portion, we've got ovules inside the ovary. If we zoom in on an ovule, let's find a good one. It would have the different cells with the synergids and the egg and the antipodal cells. And you do need to know the functions. You don't have to recognize which ones are which, because again, we're not going to have a practical portion with microscopes. You might get something with a cartoon diagram, but not the microscope ones. This would be looking inside the anthers. So inside the anthers would be the pollen and the sperm. And remember, there's going to be 2 sperm per pollen. Some of these are still dividing, so that's why it looks like there's 4 in some of them. And once the flower is ready to release its pollen, there will be an opening to let the pollen out, to disperse. Could be wind pollinated, or it could be most likely animal pollinated. So we're going to talk about pollinators as well. That one's closed, and here's a close-up of some of the pollen. Here's 2 sperm inside that one, you can see clearly. And then this is a pollen with 2 sperm and a pollen tube. So the pollen tube moves down the style after the pollen lands on the stigma. The 2 sperm will head down, so in this one they're heading down the style to the ovule, and double fertilization will occur. So fertilization and pollination are different. So pollination is where the pollen makes it to the stigma and then down to the micropyle, versus fertilization is when that sperm gets to the egg and the polar nuclei. All right, here's another one. There's the pollen, here's the sperm heading down that pollen tube. And I'm going to show you the slides for the leaves, because we're going to go outside, and I'll just tell you some adaptations for different kinds of leaves. We won't see all these kinds of leaves while we're outside, but just so you know. This one here would be a xerophyte. A xerophyte means less water, and you can tell by thicker cuticle, so that's protecting the plant from water evaporation, as well as the fact that the stomata are on the bottom with crypts. Sometimes they're hairy crypts with trichomes, sometimes they're just crypts with the stomata up tucked away, so that as the wind goes past the leaves, there's less transpiration, less water loss. If the stomata is up there. So you guys remember stomata from your Bio 230 class. So let's get to a hairy crypt. So in this one, here is the crypt with the trichomes. The stomata are up here with the guard cells. Again, multiple layers of the cuticle lets you know that it is a xerophyte versus, let's get to a hydrophyte. This is a hydrophyte. And a hydrophyte has plenty of water. Sometimes it is on top of the water, like water lily. So it would have stomata on top, because if it was below, there would be great gas exchange. So the stomata will be on top, the cuticle is very thin, usually just a layer, and sometimes there's big air pockets because this is going to float on top of the water. So again, you don't have a practical, so you don't have to worry about identifying this, but we'll be talking about the difference in leaves outside. This is just to demonstrate how important bees are, because we are going to talk about pollinators, and if there were no bees, this is Whole Foods did this display of the fruits and vegetables that are on display that are bee pollinated versus what would be gone if the bees go extinct. So we will talk about different kinds of pollinators. I think that's the last of the slides.